Welcome along, everyone. This is Humanly, and my name's Daniel Roytas, and today I'm joined by Dr. Mark Bailey. G'day, Mark. How are you?
Daniel, I'm doing really well, thanks. Thank you for having me on your show.
No worries. I spoke with your wife, your lovely wife, a couple of weeks ago, Dr. Sam Bailey, and we had a great chat. And I've been following her work for a couple of years now, and I've heard her mention that she's got a amazing partner doing some research and writing and investigation in the background for her. And I always wondered, like, oh, I wonder if he's on board with her position. And you were sort of saying before we started recording today that you were on board, and it's really great to have you on the podcast today. I can pick your brain about a few things I've got questions around. So welcome.
Thank you. Looking forward to it.
So maybe to begin with, just a brief introduction, just who you are and what your journey has been like, maybe throughout your career as a doctor, because I know you left medicine well before the whole pandemic thing. So yeah, would you mind just giving a quick sort of rundown of your history?
Sure. Yeah. Well, based here in Christchurch for New Zealand for pretty much my entire life, along with my wife, Sam, who you mentioned. And yeah, so I trained traditionally as a doctor at Otago Medical School. And I finished my degree in 1999 and then commenced work as a house surgeon after that. And I had a couple of years working in the hospital. But to be honest, the medical system never really gelled with me from the start. I sort of had a few speed wobbles along the way. And the first was during medical school when I started reading material that we weren't supposed to read. And I guess now I recognize it as terrain theory. I recognize it today of what the mainstream would call anti-vax literature. But, you know, I started reading this stuff as a student, and it piqued my interest. And I thought, it certainly wasn't as crazy as what the doctors that were teaching me were saying. They were sort of saying, don't look at this material. These are quacks, crackpots, etc. But that was the first time I sort of opened my eyes to this wider world, that there was more than just the medical models to explain human health. So yeah, and sort of as I got towards the end of my medical school training, I was also a very good athlete. Like as a junior athlete, I'd represented New Zealand in running, like in the New Zealand cross-country team. And so I had that going on the side as well. And after about 2 years of being a house surgeon, so my first 2 years working, I decided to focus full-time on being an athlete. So I became a professional duathlete, so running and cycling. Like triathlon, but in the bike run format. I could do triathlon, but I was never quite so good at swimming. But yeah, I had 5 years basically as a professional duathlete. Absolutely loved it. Learned an incredible amount about the human body, like how far you could push it, the kind of training loads that you could put your body under, maybe when training loads got too much and the body started to decompensate. But a fascinating insight into human physiology and an interesting personal experience, particularly when, you know, doing sort of 7-8 hour training days on the running and biking. It takes you to some really interesting places, to say the least, both physically and spiritually. So yeah, that was an interesting kind of chapter. But during that 5 years, it was, it wasn't like I got out of medicine completely. Like I kept my practicing certificate, and I worked usually 2 days a week over most of that 5 years, and worked in everything pretty much, mainly non-surgical specialties, but any kind of specialty you can imagine, from psychiatry to oncology, hematology, nephrology. Just worked in absolutely all of them basically to get a really good rounding of what the various specialties did. So 2007, I turned 30, and by then I'd sort of had enough of doing the full-time sports. partly because I think I'd hit pretty much peak physical condition, and I'd got into the top 10 in the world a few times, but I found, to try and get into the top 3 was just starting to get a little bit frustrating, to say the least. So I thought at age 30, I'd had enough, and it was actually the same year that I met Sam. So it was a good time to kind of stop the 7-8 hour training days and spend time with Sam. So that's when we first came together, and we just hit it off from the start. We kind of I guess you could say, I guess I corrupted her with some of my alternative medical stuff, because at that stage she had really only just started working. She'd just been in practice for a year and a bit at that stage, and I sort of opened her eyes to the fact that there was a lot of problems with medicine, and that a lot of the treatments we were giving were probably more harmful than good, and that there was this alternative world of other models involving human health and treatments, etc. But the two of us, we worked really close together when we did hospital medicine, and got married in 2009. And soon after that, we both moved into private practice and did various different things. I was mainly at that stage concentrating on musculoskeletal medicine, and started doing postgraduate qualifications in that, and also specialty exams. And I almost got to the point where I was a consultant in musculoskeletal medicine, and then decided that, I really didn't want to do medicine any longer. So for the last couple of years, I continued to work part-time clinics a few days a week. And when 2016 came along, I decided that was it. I just closed up my clinical practice, and decided that I was going to get out of medicine completely, which I did, and thought that there's no way I'll get involved in any medical matters ever again. Until and then had 3 years great time doing other things, spending a lot of time with our kids, working with my brother who did property developments. So we did that for a few years, just building residential complexes and running a few different investments, and really just enjoyed my time doing something completely different. And then end of 2019, the whole rumors start circulating about this, new virus coming out of Wuhan. And something felt so wrong from the start. And I talked to some of my old colleagues, and they seemed to buy into the narrative, that there was this deadly virus that was going to sweep around the world. And by early 2020, Sam and I we were reading everything that we could get, all the publications, everything the WHO was saying, we were doing an analysis of, we were looking at the references. We were trying to get to grips with virology, because it was relatively, we'd been taught it at medical school, but we didn't have the depth of knowledge that was required at that point to really look at the papers and work out what they were talking about when they said they'd found this new virus. So certainly by early 2020, I was back involved again, fully immersed in medical research, basically. So not practicing, but just analyzing everything that was coming out. And this sort of coincided with when Sam set up her YouTube channel. And initially she was just going to do general topics, and she may have talked about this with you, but it was just anything, basically, something about a medication or a common health condition. But then, of course, with the whole COVID situation, she decided to make a couple of videos about the situation and share some of our early insights that maybe it wasn't what people thought it was. And obviously it created a lot of attention, and she went from having a little channel to having, hundreds of thousands of subscribers, and quickly came to the attention of the censors and the medical authorities, and the rest is history, basically, since then. But essentially, initially, I was very much behind the scenes, particularly in 2020, when I was just researching I probably spend a lot more of the time having to read all the boring papers and stuff, and then distill the information, and then Sam decides what to present to the public. Probably my habit is to get a little bit too technical sometimes and try and present something which I think is, well, this is really high level, this is impressive, but Sam will just say, we have got to present it in a way that your average person can understand. And I think she's had a lot of success explaining things like the PCR, or how they, supposedly isolate viruses, or how tests work, when a patient is sick, all that kind of thing. So yeah, and it was really, so we're doing that and had a really good recipe there. And then it was last year, actually, a New Zealand filmmaker, Graham Moffat, he was chatting with us, and he said to me, why aren't you giving out in public?
Yeah.
And I said, no, look, it's Sam's channel, it's her thing. And he said, no, I disagree. He said, I think seeing you today, I said, you need to show up and show that you're right next to Sam during this whole process. And initially I must admit I was skeptical, but it was such a great reception when I started appearing publicly more often, whether online or doing talks here in New Zealand to live audiences. And it was right from the start. I was really surprised. Got such a positive response. And so many people said that they were so happy to see that Sam and I were doing it together. Because like you were saying earlier, you always start to wonder, oh, is the husband or the wife going to be on board with what they're doing? And is this causing total chaos? But I mean, for us, like we absolutely love it. And to us, it doesn't even feel like work. And I think for the first time for both of us as doctors, we feel like we're really just following a path of truth, and we're really able to do exactly what we want to do. And we realized that when we were inside the medical system and, getting practicing certificates and all that, the compromises that you have to make are just too much, basically. The truth goes out the door, and you're expected to follow a whole lot of guidelines, which often have nothing to do with the best evidence or the interests of the public. So, yeah, I guess that's a sort of a long-winded story of a bit of background.
No, it's great. It's a great overview of your experience and your journey to get to this point, because it's not often you'd hear someone with your sort of credentials questioning the fact that a virus may not cause disease or may not even exist in the first place. So I think it's really important for people like yourself to speak out. And I want to say thank you for doing what you're doing. I think we need more people like you and your wife doing these things. For humanity, I think it's really important. And was there I know you said when you were in uni that you were sort of looking at terrain theory a little bit and sort of questioning things, but was there a straw that broke the camel's back for you in like early 2020 when you're like, hang on a second, there may not be a virus? Or were you sort of aware that there mightn't be a virus even before the whole COVID thing? Like, how did that happen for you?
Yeah, I mean, to be honest, up till 2019, like the majority of people, and certainly like doctors who are trained in the system, I just assumed that viruses had been proven to exist and proven to cause disease. Now, I was shown, obviously during my training, I was shown pictures of what were said to be viruses under the electron microscope. I was shown various tests, like antibody tests, that you could do. You could order these blood tests on patients, and they were said to prove that the patient had the virus or had been exposed to it. Obviously, I thought there were vaccines that were based on viruses, because the packet said live virus or attenuated virus. And I thought, okay, well, the virus must exist if it's written on the side of the packet. But yeah, early 2020, I think it might have been actually 2019. I was listening to a podcast, but it was to do with mainly financial stuff. And the host said HIV doesn't exist, and he just threw it in there. and I had a lot of respect for this podcaster, because, he was giving really good advice about financial matters, and I'd been really impressed. And I thought, that's a bit odd that he threw that one in there, and I just dismissed it. And then about 2 weeks later, I think it must have been about November, December 2019, someone else said it as well. And this guy was like a PhD doctor, and he said it's one of the biggest hoaxes ever, HIV. And that really, I thought, this is crazy. This is two people in 2 weeks have said the same thing. And to be honest, I didn't even know that was a thing, questioning whether the viruses existed. So I started looking at the literature, and of course, all the mainstream stuff says, no, that's just crackpot theory. But I thought there must be something out there and someone that's written more extensively about this topic. And that's when I found Virus Mania, which was at that stage the second edition, written by Klaus Conline and Torsten Engelbrecht. And so I ordered the book straight away and read it in like a day when it arrived, and it absolutely caused a paradigm shift.
Wow.
And some of the most influential stuff in the book was from the Perth group, based in Australia. And I saw the arguments that they put forward. And as I say, I didn't even know that questioning the existence of viruses was the thing. And it was it seemed so exciting to me that this is a rock that needs to be turned over and examined. So it was early 2020, and that's when I said to Sam, I said, sit down, I'm going to tell you something crazy, but I think that these viruses, I'm not sure about all of them at this stage. I said, I think some of them don't exist. And I said, HIV seems to be one, the particle itself doesn't exist. I said, it's nothing to do with AIDS, that's real, but the HIV particle doesn't exist. And I said to Sam, you've got to read this book, Virus Mania. And so she read it and had a similar kind of paradigm shift, and thought, both of us could see this whole other world that needed to be explored. And so what was funny is that Sam started making videos with her new knowledge, and a few months later, Torsten Engelbrecht, one of the authors, gets in touch with her and says, wow, it looks like we're on the same page here.
Oh, cool.
Yeah, amazing what you're coming up with. And Sam said, well, it's because, we've read your book, and, that's why we're on the same page. And they just got talking, and within weeks, Sam was brought on board as a co-author for the third edition, which came out in early 2021. So yeah, that was kind of how we got exposed to the possibility that there is no viruses. And I think since that time, we've done all our own original research, and I guess every single time we look for the evidence of a pathogenic virus, we can't find one. So I guess my position is that I can't categorically say that pathogenic viruses don't exist. What I can say is that there's no evidence that I can see that they exist. So either we have to refine the techniques that are being used, the current methodologies, which are completely unsatisfactory, or we have to look a bit harder. But to be honest, I think it's unlikely that there's any pathogenic viruses on the planet. I think what's happened is that there's been a lot of mistakes with misattributing various findings, whether they're pictures or molecular detection techniques, antibodies, people getting sick, all this kind of stuff. I think people have just come up with the story that it's a virus causing all of these things, when in fact, when you examine all the evidence, you can find, you can get all of these findings without a virus. You don't need one in the equation. So yeah, I think that's where we're at. We're still open-minded, because I think it's foolish to close down the possibility of something. And as scientists, that's what we should do. We should always say, well, maybe it's possible. So we should always allow the door to be open. But I think it's looking incredibly unlikely that there are any viruses. And I think, unfortunately, what's happened is that the proponents of viral theory have drifted so far off the scientific method now that it's really hard to pull them back out of the hole that they've fallen into.
Yeah.
And if you actually look at what the scientific method involves. It involves absolute skepticism about your results. You should basically publish something and then say to everyone, look, this is probably wrong. Please pick holes in it. Tell us where we got it wrong, how we could improve it. And people should try and replicate exactly what they did to see if they got the same results. But we simply don't see that anymore. What we see is a paper gets published, and we saw this coming out of Wuhan. Fan Wu publishes a paper and says, hey, I found a coronavirus, it's a brand new one. And then instead of people saying, well, that's probably unlikely, let's go and repeat the test and do it again, everyone else takes that paper and says, well, that's been established, so let's use that as our baseline, our control, and start doing our own thing over here. And it's not the scientific method. And it's almost like become, scientism and a cult, what's going on here. And I think, as you well know, those of us that point out that there's problems with their methodology and their evidence, their reaction is not a scientific debate. It's a smear campaign, ad hominem attacks, and anything but science.
Yeah, absolutely. And you bring up some really valid points there. And I sort of want to elaborate on a little bit of what you've said. So what is the method that you would like to see? What experiment would you like to see done to show that a virus causes disease? And what are the gaps in this experiments that have been done to say there is a virus that make you question the existence of pathogenic viruses?
Yeah, well, first of all, what we need to start with is what the definition of a virus is. And it is surprising how many times you ask someone, even a doctor, what a virus is, that they can't actually answer. And they give you these kind of roundabout explanations like, oh, it causes smallpox. That's causes measles. Or they say it's an infectious thing. But they don't actually give you the formal definition. And that's, it's a tiny particle, usually in the nanoscale, something like 100 nanometers in terms of its diameter that it's supposed to be. It's supposed to have a genome, the genetic code, which can be RNA or DNA. It's supposed to have a protein type coat surrounding it. And it has to be an obligate, so it has to be an intracellular parasite. So it has to be able to not only get inside a host, but then be replication competent, and more copies are made within the host cell, and then it comes out of the host cell and infects other hosts potentially and causes some disease. So I think you need to show all of those steps. You can't just show bits and pieces of them and say that you've proved that there's a virus. You have to do, absolutely all of them. So in terms of how we would do that, so first of all, we'd have to find a sick organism, whether it's a plant or an insect or a human. And we'd have to say that it's got a particular illness that we can recognize as particular features. Then we have to try and isolate the apparent virus from that organism. Now, ideally, you'd just do that directly from the organism itself. So you'd just take a sample, maybe like a blood test or saliva, and then you'd potentially process it with a centrifuge or something like that. But you'd try and find the particle inside the organism that has the disease. Now, unfortunately, that's never happened. And that caused major problems in the 1900s when the electron microscope was invented in the early 1930s. They thought they'd just be able to take samples from people who were sick and then find these alleged viruses inside them. But unfortunately, that didn't really work out for them, and no matter how hard they looked. And I think a lot of the public don't understand that, is that it doesn't matter how sick a person or an animal is, they never find a virus inside that person, directly from the person. And this is even people that are riddled with diseases like AIDS, and supposedly they've got this huge amount of HIV in their system. And yet when you take a blood sample you can't find one. You can't find any of these particles. You just don't see them. So that's that of course is a major problem, and it means that you can't get past step one of Koch's postulates, which is why you often hear people say, well, we can't do Koch's postulates for viruses. I mean, technically you could, it's just that they've never got past step one, and that's why they don't want to do anything more to do with Koch's postulates. It's because of that reason. So the next step, because they're unable to do that, they resort to cultures, what they call cell cultures, which realistically should be called tissue cultures, because it's just a whole lot of biological soup, basically, with some sort of host cell in there. But again, there's some major problems with the techniques they use to do these cultures. One is that they often use cells which are already abnormal. So they might be cancer cells, they might be like Vero cells, which are monkey kidney cells, but they are genetically abnormal. They're not normal cells. And often they're not from the same species that you're, testing this alleged virus on, which is problematic too. And then they're often not from the same type of tissue. So you might say this, made up virus SARS-CoV-2, it's supposed to affect human respiratory cells, like lung tissue, but they end up using abnormal monkey kidney cells to do their experiment. And you think, well, what's that got to do with, it's a different species and it's a different organ. But the reason is because those types of cells are more reactive in the test tube. So if we took cells from you, for instance, and you're healthy, your lung cells aren't going to do anything because they're just too resilient, basically. Right. So whatever mixture you add to them in the test tube, you don't get all these breakdown reactions, which they end up attributing to viruses. So we've got problems with the way they do the cultures. And then they, you probably looked into some of the stuff, but not only do they have their monkey kidney cells in there, but then they add a whole lot of other stuff. Like they add cow's blood, they add antibiotics, antifungals, various other types of chemicals. And then if that doesn't work, they passage, the sample, which is when they take out a small amount of the cells and then reintroduce them to more antibiotics and more cow's blood and more toxic chemicals. And they may do that several times. And eventually, if they see some sort of reaction and cell breakdown, they say, well, that must be the virus. And the problem is that it's been shown that most of the time they don't do valid control experiments. And sometimes they say they did a mock experiment, but you actually, if you get in touch with the authors and say, what did you do? It's often not a valid control. Like they didn't add any biological material from a human, say. They'll put in their kidney cells, their antibiotics, their cow blood, etc. But then they don't add any human tissue. And really that's the key, is that if you're not doing that, you don't have a valid control experiment. And probably there should be two controls. One should be a human sample from someone who's completely well, and another could be from someone who's unwell with a similar condition, but said not to be from the virus that they're trying to investigate. So, but as I say, they never kind of do these valid controls. And we know now that you can simply do those culture experiments, and just by stressing the cells in the tube, eventually they break down, the so-called cytopathic effect, and they start producing vesicles. And sometimes at that point, they take some, electron micrograph images and say, well, there's the virus. It's coming out of the cells, or it's in the cells, and it caused it to break down. But I mean, simply taking pictures like that doesn't prove anything. I mean, all we're looking at is vesicles, and they look exactly the same as other vesicles that come out of mammalian cells. And what I find deceives people is that somebody puts an arrow or a circle on one of these bubbles and goes, that's the virus. Yeah. And it's bizarre, because you're sitting there going, I don't understand how they're claiming that particle came and destroyed the cell. For all we know, it's just a vesicle doing what they do. So I think that's a major problem. And then you get into the issues of other indirect techniques like the genetic stuff, finding genetic sequences. One way is just through PCR. But again, that tells you nothing because you don't know where the genetic fragments came from. So, they've got a test tube and it's full of all this material. Nobody managed to purify anything or demonstrate where the genetic material came from, but they simply run a PCR or do genomic sequencing and then say, wow, that came from the virus. But again, there's absolutely no proof of that. So, so to this day, if we look at all, viruses that are alleged to exist, we've got the same problems of no one's actually demonstrated that it's the particles by themselves that are causing the cells to break down, that are causing disease in other people, or are able to transmit from person to person or animal to animal. Instead, we've just got various indirect kind of techniques. So to come back to your question of what we actually need to do if they're going to tell us that there are pathogenic viruses, is that if they can't find them inside people, which is a bad sign to start with, then they've got to do one of these culture experiments, and then they have to take those particles that are coming out of those cell culture experiments, and they've got to properly purify them. Now, there's various ways to do that with filters, but usually with centrifugation. Like if you do a density gradient and you try and get the particles purified in a band. And you can confirm, this is where electron microscopy is good, to confirm that you've got purified particles, that they all look exactly the same morphology, same size, and then take those purified particles and demonstrate what they can do. Like, spray them in the air, spray them into an animal's cage, or if you're doing a human experiment, just, spray them in our face and see if those particles by themselves do anything. But I think there's a problem. They don't do anything. And that's the whole issue. So that's why I feel there are no pathogenic viruses, because I think they would have done those experiments and they would have been, so easy to do, basically, that all we see now is a whole lot of excuses as to why they can't do them.
And it's not like they haven't tried to do those experiments where they've taken a sick person and put them into contact with a healthy person, or even taken the biological fluids from a sick person and exposed it to a healthy person in every which way possible and made that healthy person sick with the sick person's disease. They have tried to do that and they've failed. And I don't think a lot of people realize just how desperate they were getting, especially in the early 1900s, with a lot of these experiments trying to infect people with measles and chickenpox and influenza, and they couldn't do it. So that in itself would sort of make you question, without even going and doing all these cell culture experiments, that hang on, maybe this isn't the way that a virus is transmitted, or what we observe as contagion.
Absolutely. And this goes deep, because it's not just viruses, it's all microorganisms. And I think we have to be clear, we're talking about, like you said, contagion, in the sense of it's going from host to host via a microorganism, whether that's bacteria or a made up virus. And the clinical experiments, they you never get ethical approval to do them these days.
Not these days.
No, you've probably seen some of them. They do wacky stuff, like, injecting blood out of one person into another person, taking various biological fluids and pouring them into people's throats and spraying them in their eyes, all sorts of stuff, which couldn't really be done these days. And you're quite correct. They tried with so many different diseases, but couldn't. Influenza is the classic one. If you look up, of course, there's the 1918, the Rosenau experiments were really famous, with 100 volunteers, couldn't transmit one case of flu to these 100 healthy volunteers. And since then, there's been a few other flu transmission studies that they tried to do, which again failed 100% of the time. And what's interesting is, if you read anything about influenza, they'll say that it's highly contagious, but then strangely don't mention all of the studies over history, which have completely failed to, to show that it is transmissible in that fashion. We find it fascinating because we've got whole textbooks on influenza, and it's really amusing because not once do any of them mention all of these failed experiments, and instead they look at indirect things, and they'll talk about, oh yeah, in 1976 there was such a bad outbreak of influenza and it spread so quickly, and these were the R values and all this kind of stuff, and you realize it's all complete rubbish because they never demonstrated transmission at all, basically. And similarly, when people say things like chicken pox and measles are highly contagious and transmit via the aerosol route, and you'll see some claim that even being in the room with the person for 30 seconds could put you at risk. But then you look historically at the studies that they actually did, sometimes with children, sometimes with monkeys. They never demonstrated any aerosol transmission. Typically they'd take, if one animal or child had some sort of vesicles on their skin, they'd aspirate some of the fluid, and then they'd go and inject it directly into another subject, and then get some sort of skin reaction, which sort of looked like the donor's skin reaction. And sometimes they wouldn't get systemically unwell, so no fever or anything like that. And then they'd say, well, that proves that there's a virus and it's transmitting. And you think, well, that's not, nature doesn't do that. That's ridiculous, having to take the fluids and inject it into another subject. And we see the same Mickey Mouse kind of stuff going on with animal experiments in general. And, we saw that with SARS-1, where they took monkeys and just poured biological muck into their lungs, and lots of it, like relative to the size of the monkey. And I mean, it's just absolutely stupid, because if you pour these liquids into any subject, you'll get reactions, like they will cough and they may get some lung damage. But typically they never get the same symptoms that the disease is supposed to come with. It's and I think it's a real problem, because when I trained as a doctor, I was never taught any of this stuff. I got given this version microbiology book that just had chapters about measles or chickenpox or HIV, and they just give you this version that it's a very infectious particle. This is what it looks like. This is how many people have got it. This is how you treat it. There was nothing in there that said, oh, and by the way, there's actually never been a transmission demonstrated once in history. You'd think that would be kind of the key point, but strangely absent.
They don't tell you that, yeah. And do you think it's the same for bacteria as well? Because obviously there are diseases that are caused or are said to be caused by bacteria. Say things like scarlet fever, for example, was a bacterial disease which seemed to just vanish into thin air. What's your perspective on bacteria and disease?
Yeah, I mean, we've got the same problem of just them not being able to demonstrate that there's contagion. Now, with bacteria, we can certainly identify them and we can isolate them properly, purify them. We know their composition, we can do experiments with them, etc. So we're in a different category now than viruses, which are hypothetical and seemingly non-existent. But even with bacteria, if you look up, I mean, one thing we found really interesting was looking up Koch and Pasteur, the famous germ theorists, the fathers of germ theory, and actually looking at their original studies. And you probably know Pasteur's work is just highly fraudulent. He was just making stuff up, essentially. But even with Koch's work, some of it, I was like really surprised at how it didn't really demonstrate contagion. He'd have these animals in cages for months and then say that they got tuberculosis off each other. And looking at the experiment, you thought there was no kind of control experiment, like putting an animal in a cage in a laboratory and then giving it various stresses is going to make it sick, basically. and you haven't established that the primary cause of the disease was a bacteria coming and, jumping into their lungs and making them sick. So, but I don't think that's appreciated by the vast majority of the medical profession, certainly, because they just read the microbiology books that they get given and they go, oh, here's Koch's postulates, and that's how you work out whether a microbe causes disease. But they don't say that wasn't really done properly, and that the control experiments weren't done in those days and haven't been done ever since. And what they tend to find is associations. So, you'll see something like, whooping cough is a classic, because, they say that the disease is caused by Bordetella pertussis, this very specific bacteria. But then, they get into trouble because then you find more sensitive ways to check for the presence of Bordetella. And one of those ways is PCR. And then and PCR in this application definitely has problems for diagnostics, but it probably can detect whether there is Bordetella if you do a good PCR design. Right. And what they find is that lots and lots of people have Bordetella in their lungs, but they're not sick.
Yeah.
And there's many people like myself. I've never had some sort of coughing illness. Like some people seem to get these really bad coughs and stuff. But, in my lifetime, I've never had anything like that. And I'm inhaling all the same microbes as everyone else.
Right.
And no doubt I've got some Bordetella in my lungs, or have at least had them in there before. But there's no way I'm getting whooping cough. And it's nothing to do with vaccines either, because we know that despite the fact in countries like ours where they vaccinate a lot of people for whooping cough, people still seem to get it, But my point is that the presence of the bug doesn't do anything. And probably most people are walking around with small quantities of what are said to be pathogens. But clearly for the vast majority of people, they don't do anything. And I know that you've looked into terrain theory, and you know that in order for a microbe to appear to be a pathogen, there has to be an underlying change in the host tissue. So in the lungs, that could be like a toxic insult, whether it's a really cold air or something, which actually physically damages the lung. It could be chemicals like smoking, air pollution, all that kind of stuff can damage the lungs. It could be a malnourishment, vitamin and mineral deficiencies, which cause the lungs to malfunction. It could be another form of toxicity in the body, which tries to clear itself through the lungs and creates a whole lot of dead tissue that's trying to get out. And in all of these situations, you're going to get a potential proliferation of microbes, but they didn't cause the problem. They're just reacting to the problem, and they're doing what they do, which is gobble up dead tissue when it appears, because if they didn't do that, life on our planet would be finished. It would be over. I mean, we need this. We need dead tissue to be reprocessed basically, and put back in the system again for more life to go forward. But I think it's the biggest misunderstanding of, and I think it's a paradigm shift. And I don't know if this has happened for you, but you go from thinking that, bugs are a bit of a problem, you've got to be careful, they might get you, to appreciating that it's all driving life forward and there's no bad bugs or anything like that. They all just have different roles to do depending on what's happening. And, if someone says that they've got a bacterial infection, it sort of implies that the bacteria has caused them this problem, whereas you can always find that something went wrong first, and then the bacteria moved in or proliferated and just did their thing. It's confusing too, because sometimes if someone gets really sick, they can get a huge proliferation of microbes, and the microbes themselves do produce toxins sometimes, which can be deadly. So there's confusion with that as well, of people who say, well, no, but actually the microbes are really dangerous. And there are certain situations, like if you have a lot of tissue damage, then the microbes themselves may start being damaging, but they're certainly not. They didn't trigger the whole thing. They didn't cause the person to become unwell.
Yeah, those microbes are coming to clean up the mess. And by the very virtue of metabolizing dead and dying tissue, you will get symptoms associated with that, which is the healing process. And as you said, they might produce toxins, which could then make you sick from that metabolic process of cleaning up the dead and dying tissue. And I was reading a paper the other day where they were looking at heavy metals in milk, and they were showing, I don't think this was actually in humans, it was in vitro, but they were showing that certain types of E. coli would proliferate when you put, I think it was mercury into this tissue bacterial culture. Certain types of E. coli, which are native to the human gut, would proliferate and digest the heavy metals. So if someone had gastrointestinal symptoms, you do a stool sample or fecal sample and they've got an overgrowth of E. coli, say, oh, it was the E. coli's fault, but actually it might have been because you were drinking some heavy metals in your milk and now you're getting sick from that. So we're actually blaming the bacteria, which are there as our friends. They're there to clean up the mess. So it's sort of a war against nature in a way, isn't it?
Totally, yeah. And, when I worked in hospital medicine, I was taught, the fallacies that you do a stool culture and find out, what's in there. And then if something seems to be in there too much, you blame that. And then you chase after it with, antibiotics. And then the patient seems to get even more problems. And then you try some other antibiotics. And, each course of antibiotics is like a nuclear bomb going off inside your body. Basically, they might claim that some of them are selective, but that's just kind of marketing spin from the pharmaceutical companies. Your body basically gets such a marked reaction to even a day's worth of antibiotics can change your microbiome for months at a time. So no, totally. It's a misunderstanding. And it's almost like a diagnostic dead end for mainstream medicine, because they think, oh yeah, we've found the problem. It's this particular bacteria, and we just have to keep throwing antibiotics until it until it clears up. When totally, and they miss the underlying cause. What was it that triggered off that proliferation or the change in the microbiome? Because nature's trying to do something. And I like the word you used. It's more like a healing phase rather than a sick phase, because I think that's the other issue with a lot of mainstream medical practice, is that sickness is viewed as something that needs to be combated with medications. But we shouldn't call it sickness. We should say, look, the body's trying to heal and we need to assist it. And often we're missing the signals, like nature's trying to tell us what to do, but we're not getting it right. And I always say to people, because it's even some of our family members are still, they get hooked up on the mainstream medical model and they go, oh, but I think this medication could help me. And I always just say to them, your body, the way it works is that it needs nutrients, vitamins, it needs the right kind of ingredients to be healthy. Those ingredients don't include chemicals. And when you swallow or have chemicals injected, there's only one reaction that your body has, and it tries to get them out as soon as it can. And I think once you appreciate that, you can understand health in a much, much better way. And if you can show people that, look, you're vitamin A deficient and your body's crying out for it, and it's in a healing crisis right now, and this vitamin will help, and the difference is huge compared to saying, well, I feel unwell, and if I take this chemical, then I'll feel better once again. But there's no way that their body was deficient in that chemical. It's not a thing, and the complexities of when these chemicals are ingested, I mean, they have all sorts of effects that we can't imagine. We've traced a lot of the metabolism of various medications, but even for really common medications, it's still a lot's unknown. about what they do once they go inside the body. And that would include things like paracetamol, which people, take around the world every day as though it's, completely safe and got no problems. But, to be honest, I can't even think of the last time I took paracetamol. I mean, like most people, I used to get an ache or a headache or something and think, oh yeah, take a couple of those. Whereas I think since having a different understanding of health, nowadays I say, why have I got a headache? And you have to be honest with yourself, and you find out why you've got it, if you're really honest with yourself, and you remedy it. You don't pop a couple of pills and then continue abusing your body. Yeah, exactly. I mean, yeah, it's amazing. I used to work with doctors who would tell me that, they'd take low sec for their stomachs, mm-hmm. and just, an anti-acid medication. And I'd say, why? And they'd say, oh, it's usually because, when I drink alcohol and it really upsets my tummy. And they said, but if they take the low sec with the alcohol, then everything seems to be okay. And you just say, no, your body has tried to tell you something. Like the alcohol or the type of alcohol or the amount is toxic. And, taking an extra medication to make the symptoms disappear is not the answer. You're going to end up with major problems down the line, and again, it's just, it's a complete paradigm shift from what we were taught in medical school, because when you graduate, you've got this repertoire, hundreds of pharmaceuticals and techniques. And then it's kind of like when you have this awakening, you sort of go, oh, most of them are pretty useless, Yeah. It's sort of disappointing, but in a way it's, you've got to get through that phase of saying, look, it's just not what it's cracked up to be. They're nowhere near. I mean, you've probably realized this too, that a lot of allopathic doctors, mainstream doctors, are not in the business of curing people, and it's not really a word that they even use. They talk about treatment and management and all this kind of stuff. And I think that was really an eye-opener as well, of going that you should aim to cure. And if you're not curing someone, you really have to ask, are you on the right track here? And I think that's another a huge paradigm shift that you have to make, of actually aiming to get people back to complete balance. And it's amazing, because in the last 2 years, and I think you've had guests talk about this too, is that you meet people who have quite advanced conditions and are really unwell, and they actually reject their own training and their own medical system that they're part of, and find a way to heal themselves. And often for them, that is their awakening, that, there's something wrong with the medical industry, because it's not able to cure people.
Yeah, and unfortunately, you have to learn the hard way to realize that medicine doesn't hold a lot of the answers to our health issues. How are we going for time? Because I know we've been going for about an hour now, and I'm aware that you've got a young family, but there are some other questions that I really want to ask you. Are we okay to keep going, or should we wrap it up shortly?
Definitely. No, we're good. I think our youngest one, he's just 6 months old, but I can't hear anything upstairs. So okay. I think he must be asleep. So yeah, no, we're good.
Awesome. So the next question is, what do you think's going on with this whole COVID-19 thing? If there's not a new disease, what do you think people are getting sick with, if they're getting sick with anything new that they haven't been getting sick with for years prior? What's your take there? I'm really interested to hear your perspective.
Yeah, well, it's where do you start with this thing? And what we've tended to do is to go back to the source material and say, was there any evidence of a virus? No. Was there any evidence of a novel condition? No. And what's happened is that to me, it's a fraudulent situation. And I wrote a paper last year with my co-author, Dr. John Bevan-Smith, and we called it the COVID-19 fraud and war on humanity, because that's what we see it as. And in that, we point out that the COVID-19 is not a definable condition. It's essentially, in terms of a clinical, set of signs and symptoms, because all it is a PCR test, or in more recent times, a rapid antigen test. And they say that's it. But I mean, in no time in history can I think of classifying an illness in that way, where it's solely based on a PCR process, which, as you're probably aware, is not really a test even. I mean, it's a manufacturing technique. It just amplifies genetic material. So, I mean, essentially, it's just a PR campaign where they've confused people, and they've said that there's this new virus, which there's no evidence for. They said that there's a new condition, which is nonsensical when you look at the definition. It's not a clinically definable specific illness. And all that we have is a rollout of PCR kits and now rapid antigen kits, which are not capable of telling anyone whether a person's sick or not. And simply, so anyone that's sick now and has one of these tests is just put down as a COVID-19 case. And it's just absolutely obscene. I mean, they're trying to claim that this has killed 5 or 6 million people, which is, one of the biggest frauds that we've ever seen in our lifetimes, basically, because not one of them has been proven to have died from a novel infection or novel virus. It's just, there's no evidence for that whatsoever. So in terms of, your question about what is making people sick, I mean, it's just all of the usual things that are making people sick, influenza, pneumonia. We've seen ridiculous cases where people are having heart attacks and strokes, and they're saying that this is due to COVID-19. We're basically, it's a reclassification of illness, but I think it's introduced more problems because you've got a state of global psychosis, basically, and that makes people unwell. And you see it all the time, the fear, the anxiety in the population, and those factors alone make people sick. I mean, we were astounded back in 2020 going out for the first time during the lockdown, and just the sense of how people just viewed you as being so dangerous to be around that they'd walk across to the other side of the road, literally, or avoid coming anywhere near you. And I mean, all these things, they make people sick. It's not a healthy way to live. So, and I think you can get into kind of waste of time arguments when, people are like going, well, prove what happened in this country and prove what happened in this city. And, we just don't have all the information. We know that in a lot of countries, it was awful mismanagement of the populations. They just did terrible things, particularly to the elderly people who were really susceptible, social isolation, not getting good nursing care, all that kind of stuff. And I mean, when we look at it, particularly back in 2020, most of the people that were dying were the elderly. Lots of countries had the average age of death of these made-up COVID cases was, about 80 years old in the developed world. So, it certainly wasn't affecting young people. So, the way I see it, and as we outlined in our essay last year, is that we're just witnessing a global fraud, basically, and there's absolutely no basis to it.
And they're rolling out a treatment for this non-existent disease now for every single man, woman, and child on the face of the planet, which is very concerning because for a number of reasons. Like, firstly, there's no informed consent. There's basically no evidence to show that these treatments do anything to save anybody. There's coercion going on, and we're told, or this is sort of sold to us as it's safe and effective, and it's the only way that you're going to be able to protect your friends and family from getting sick. But the opposite to that is true, right? You're possibly putting your health at risk by getting one of these injections.
Yeah, absolutely. I mean, there's no point getting an injection for a virus in a condition that doesn't exist. And that's essentially what they've tricked people into doing. I mean, because people talk about, bioweapons and all this kind of stuff, and they get confused because they think that it's a virus that was somehow made in a lab and released into the public and is passing around, but that's not what's happened. The weaponization is all to do with the injections themselves. So in terms of taking those spike protein sequences, which as far as we can see, they've been playing around with for about 3 decades now. If you look back to, there were patents on spike protein sequencing sequences dated back to about 1990. And as far as I'm concerned, all the research I've looked at shows that the spike protein is just a product that you can make in these these tissue culture experiments, basically. And they're a product of cell breakdown. What their exact function is, it's hard to say. But of course, they've attributed it to coronaviruses in this made-up fiction. And so what people are being injected with is essentially a weapon, because if it works, it makes their body produce the spike protein sequence and at the spike protein itself. And you don't want that in your body. It's pro-inflammatory. It's small, so it can go virtually anywhere in the body. It goes into the brain. It goes into virtually every organ. Same with the lipid nanoparticles. By themselves, they're pro-inflammatory. They're not things that you want inside your body. They're so tiny that they basically go everywhere as well. And then you've got these other constituents which haven't been declared, like the graphene type particles, which are highly toxic as well. So there's this recipe of disaster. Basically, you've got all these different constituents which can cause inflammation inside the body, and some people are getting reactions pretty quick. I think for others it may take years before their reactions manifest, particularly if they get walled off areas of inflammation inside their bodies, which could eventually turn into what they call autoimmune conditions or potentially tumors, that sort of thing. And the other thing was that, they did all their campaign to say that, these are not genetic therapies and these can't alter your DNA. I mean, I don't know that I found it hard that they even got that one out of the gate, because, I mean, they are genetic therapies. That's what they are. They're injections of RNA. And to say that they can't alter your DNA is just, I mean, that's ridiculous. We've known for 50 years that you have enzymes in your cells, and normal mammalian cells like ours, that have reverse transcriptase enzymes which convert RNA back into DNA. I mean, that's not a new concept, and it was known about. And I'm not sure if you saw, but there was a study that's just been published in the last week or so, where they took, human liver cells, admittedly abnormal human liver cells, but they put the Pfizer BioNTech injection and mixed it with these cells, and within 6 hours, the sequence had gone into the DNA of the cells. So it was like no time at all.
Yeah.
And it's absolutely crazy that I suspect they already knew this before they started injecting the populations, and I wouldn't be surprised if they hadn't already checked whether there was genomic integration. I would say they'd know that from the early Pfizer trials, because it's not that hard. In this application, we can actually use the PCR, and it's quite useful because we're looking at DNA, which is often easier to sequence than RNA. And also we're able to get a pure sample because, we can get it from a human being and say it's definitely come from a human, not a made up virus. And we could just check with a well-designed PCR protocol to see, is the sequence that they've been injected with, is it now permanently integrated into their genetic code? And I mean, it seems that there's a really high chance that it is. And, this could affect hundreds of millions of people now.
And that sort of brings me to my next question. What constitutes a human being? is it our DNA? And if we start messing around and genetically modifying, because that's essentially what we're doing, right? We're genetically modifying the DNA of people with these genetic therapies, because that's what they are. They're a gene therapy, they're not a vaccine. Are we still considered a human being if we've modified our genes?
Yeah, I guess now we're getting into spiritual and other considerations. And yeah, I mean, to be clear, I'm not an advocate of the central dogma of biology, which is, that we have a DNA code and that gets transcribed into RNA, and then that gets translated into the proteins, and it just all flows in that direction. I think when that was put forward by Francis Crick in the 1950s, I mean, he just basically asserted it. There was no,
exactly.
I didn't see an experiment he did or anything.
Yes.
And I would have thought that within a few years, it was apparent that the central dogma didn't apply. But so I think that we are very complicated in terms of our interaction with the environment, and that our DNA may change depending on environmental exposures. And it looks like it can certainly change with these experimental injections, which are a terrible idea because it's not something that we'd encounter in nature. But yeah, I mean, I think, yeah, we're dealing with now the world we're in is that the humans may have genetic edits essentially, and whether or not you still call them what you classify them as. I mean, I wrote an article recently and referred to them as GMOs because that's essentially what we refer to it if it's in agriculture or something like that.
Yeah, that's sort of where my thinking goes.
They are modified, but perhaps we should be more clear and say that they're artificially modified, because maybe some of these modifications can happen under certain pressures in nature, but in this case it's a very artificial situation, and I think it will end so badly. Like the whole promise of genetic editing and curing these diseases, from the stuff I've looked at, it's just pure fiction. They've got nothing. They're not going to be able to help anyone. It's like this ridiculous, it's like a child playing with something that's far beyond its comprehension. And I think that the whole gene editing thing will just end up in a total disaster, basically.
Yeah.
And those of us who reject it and understand what it means to be healthy and to be connected with the universe are going to be doing a whole lot better.
And with the graphene oxide, do you think that's been confirmed beyond reasonable doubt that is contained within the shots? And if it is, why do you think it's in the injections? What's its role?
It's definitely in there, I would say, because it's interesting actually. I've just in the last week had a friend do an analysis of a vial, and because it's really because you get these kind of we have secondhand accounts. Like we know people that know the people that do the analysis, and how you sort of like, you definitely trust them because these are good sources, but you always want someone a little bit closer. So for the first time in the last week, I've got a first-hand account of, looking at what's inside these things and seeing some video footage, and it's absolutely crazy. There's a few more experiments we want to do, and perhaps look at some other vaccines under the microscope as well, just to see what's inside those. But yeah, I think it's beyond a doubt that the graphene is in there. I think it's beyond a doubt that there are self-assembling particles in there. I've seen the video footage of the little particles self-assembling, but the nature of what they do, I can't comment on. Right. And yeah, why they're in there, I mean, it's, the imagination can run wild. Some people have tried to say it's just a side effect of the manufacturing process. I think that would be highly unusual, because there's a whole lot of other products that we have that don't have graphene in them. So yeah, it's yeah, I mean, yeah. Do you have any idea why they're? It's sort of like one of those conversations where you feel like you're going out on a limb because you know it could be something that's really bad.
I don't know, and that's, I don't like to speculate, and that's like sort of another thing why I don't like to make up reasons as to why I think people get sick when we observe groups of people in a location falling ill with the same thing. It's like, what makes us sick then if not a virus? Well, I don't like to just make up anything, because that's how we got here in the first place, is we just said, oh, it's a virus, we just make it up. So I don't want to say the same thing with like graphene oxide and then later on down the track regret things that I've sort of said to lead people astray. So I've got ideas, but I think we just have to wait and see what research sort of comes out there. There was I think it was a German PhD scientist talking about graphene oxide a little while ago, and he was saying that they're very like thin, like filaments, and they're like razor blades, and they can destroy the inside of the like the endothelial lining in the blood vessels. And I was thinking, and I asked this to Dr. Kaufman when I had him on my podcast, is it possible that those razor blade like substances are destroying or damaging the endothelial tissue, and the spike proteins that we're seeing are just little bits of protein from the endothelium? Is that a possibility?
Yeah, I mean, I think there's two different things going on. I think the mRNA is coded to produce the spike protein, and I presume that's happening in most people. Although, yeah, I have heard Andy Kaufman question whether people are even producing this spike protein at all. But other researchers say that, yeah, it's definitely being detected inside people even months after they've been injected. I think the real danger with graphene is, like you say, it's so unusual in that the graphene plates can be 1 atom thick, and your body's not designed to process. It's not something it would ever encounter in nature. And yeah, we were looking at quite a few papers recently for one of Sam's videos, which showed that these graphene particles can just go straight through a cell, like straight through human cells, as though there's nothing there. And I mean, they can also cut straight through genetic fragments. Like, the plates fit between the nucleotides in a genetic sequence. I mean, that's how fine they are. And also at that level, they behave in all sorts of strange ways. And as you know, graphene's got unusual properties with conduction and the way it responds in various electromagnetic fields. So yeah, I mean, I've seen a few theories that it's the graphene itself that is causing a lot of the heart problems, but not through a cutting or lacerating type mechanism,
right,
but through conduction issues. Because we know that if the graphene is in a biological entity, it will gravitate towards where there's electrical activity. So in a human, that's going to be the heart and the brain are going to be areas where there's a great deal of electrical activity. So that may be, I've seen some people postulate that that's why we're seeing a lot of people have heart problems, is because the graphene is interfering with conduction in the heart and causing problems. Because I must say, I mean, a lot of people I know have had heart problems. It's just a question of what exactly is the mechanism. Is it an inflammatory one through the spike protein, or is it the graphene particles themselves causing inflammation, or is it the graphene particles causing conduction issues with the electrical system? I just think it's so many unknowns, and we've been a little bit more reserved on that stuff because unlike going back and looking at the history of virology and measles and coronaviruses and that, where you have just so much in terms of research that you can read through and experiments you can check and stuff. The whole thing with the constituents of these injections is that it's just all new. And yeah, a lot of it's speculation at this stage. But yeah, I think there's just so many things in these injections which appear to be incredibly dangerous that, yeah, I mean, no one in their right mind, if they know anything about them, should be getting them injected into themselves.
Yeah. Yeah, I appreciate your perspective on that. I also wanted to ask you, do you think medicine's going to be able to redeem itself after all of this?
I think it's yeah, I know we talk about this too, because within New Zealand, there's hundreds of doctors that have really got out of the system or are half out of the system now. And a lot of them feel that no, there's just either the whole thing's going to collapse under its own fraud, or we're just going to go into different paradigms where we're going to have a medical system which is even more based on centralization, you know, pharmaceuticalization, if you want, for want of a better word, you know, requirements to comply with the system, like, you know, you can't access certain medical care unless you've done certain things, like have certain injections, etc. And we may just get a splitting of the system where many people realize that that's a toxic system and it's not the way forward. And already in New Zealand, there's parallel health organizations that have set up and are looking at much more holistic models that are not reliant on government funding, that are not reliant on pharmaceuticals so much. So I think in some ways we should look at it as a really good opportunity. And I think for a lot of people, the last 2 years has been a huge awakening, not only for medical practitioners and medically trained people like ourselves, but also for the public in general. And people need to decide which kind of model they want to get involved with. And I think for a lot of people, they've realized that the medical system is not the way to go. And I've never seen, like in the last year, so many people just contacting us saying that they are so dissatisfied with their doctors now. And, you know, they're deregistering from practices or just not even bothering going to see some of these doctors anymore. Yeah.
Yeah. It's going to be interesting to see what happens and watching that scenario play out. And sort of moving forward with what's happening with all of the pandemic issues and healthcare and government overreach. I mean, it's unfortunate that it happened, but in a way, as you said, it's a necessity and it's allowed a lot of people to wake up. And I think it's been a blessing in disguise. What do you see the future like in the next 18 months? Do you think there's hope for humanity? I've still got hope for humanity.
Yeah. And do you know, for both Sam and I, we have this very much, we're into a very present kind of mentality. Like we don't speculate about what might happen in the future. So I don't usually try and make predictions. We just usually focus on what's happening in the moment and doing the absolute best we can to be conscious and bring understanding and enlightenment into what's happening in that moment. I mean, for sure. I mean, humanity is going to win. We just don't know when. And I'm not too worried, you know, if we go, you know, we've had this has happened many times in history where we've gone into dark periods, and sometimes they can last for centuries and everything goes backwards. But over time, you know, we get there and we get, the consciousness keeps rising. And I think things will definitely in the long run. I mean, it's really optimistic. I mean, we're certainly optimistic because, you know, we keep having kids and, you know, and our kids, you know, they can see our optimism and, you know, thriving at the moment. So that's great to see. So, yeah, I mean, I'm a great believer in you just create your reality and truth. So if we remain motivated and positive, we're heading to a good place. And I think if we get despondent and give up, then that's what's going to happen. It's going to, you know, things are going to look awful. So, yeah, I mean, in some ways, we've just whatever happens at the moment, we just look to respond and lift our game even further. So like you, I see it as a great opportunity the last 2 years. And no, I'm grateful for what we've been given, and for everything really that's happened, both in terms of our own personal awakening, and as well recognizing a greater awakening in this community around the world. So yeah, optimistic.
Good. I'm glad to hear that. And I like that you live in the moment. And you're right, we do create our own reality. Any final words, Mark? I'm going to let you get back to your family, but if there's anything that you wanted to add that you feel like we haven't discussed, or any sort of final concluding remarks?
No, I think we've covered a good amount there, and I really enjoyed talking to you, and I'll keep watching your podcast, because you've had some amazing guests on, and I look forward to continuing this journey with you.
Thanks. Yeah, it's good to know that we've got good, strong, well-educated men like you on our side. It's always refreshing to meet a new face and connect. Now, where's the best place for people to stay up to date with what you're doing? So I know you do a lot of writing for your wife, Sam. So would it be through her website, or have you got your own website? Or
yeah, no, definitely. It's enough work just keeping up with all Sam's platforms. So yeah, I publish my work on her platforms, and so the best one to go to is drsambailey.com. So drsambailey.com. And of course, she's got her YouTube platform, her Odyssey platform, and Telegram as well.
Wonderful. And I'll be sure to link all of those websites in her Telegram channel in the show notes as well, so we can get more people following your good work. Thanks so much for coming on, Mark. I really appreciate it. And it was an absolute pleasure speaking with you.
Thank you, Daniel. It was great.
Thanks again.